Have you ever just stopped and wondered what's really swimming around in a single drop of pond water? Or, what the actual architecture of one of your own cells looks like up close? For almost all of human history, these worlds were completely invisible, totally locked away from us. Welcome to FZ-LabTech channel. Today, we are going to go on this incredible journey to explore the history of microscopy and the absolutely ingenious inventions that let us finally see the unseen. So it really all boils down to this one question, right? How in the world did we go from seeing only what our naked eye could see to visualizing the very building blocks of life itself? It's a fantastic story of curiosity, of optics, and some truly brilliant breakthroughs.
Okay, so our story begins centuries ago, with the very first attempts to bend light and make small things look, well, big. And this wasn't some single eureka moment. It was a slow, fascinating evolution of an idea.
This timeline really shows how each discovery was a stepping stone for the next. It all started pretty humbly back in the 1590s when a couple of spectacle makers, the Jansen brothers, just kind of put two lenses in a tube. Simple. Decades later, Robert Hooke uses a similar gadget to look at cork, and he gives us a word we use every single day now: cell. Then things get really wild. In the 1670s, Antony van Leeuwenhoek perfects these little single-lens microscopes and uncovers the shocking, teeming world of what he called animalcules, what we now know are bacteria and protozoa. And then things kind of stalled for a bit. For over 200 years, progress was pretty slow. Until 1931 and the invention of the electron microscope. I mean, a true quantum leap that changed science forever. So, these early pioneers could see stuff, but they hit a huge wall. How do you actually observe living cells when they're mostly water and almost completely transparent? It's kind of like trying to see a perfectly clear jellyfish in a glass of clean water. It's tricky.
And this slide just illustrates the problem perfectly. On the left, using a standard bright field microscope, those living cells are just faint, ghostly outlines. You can tell something's there, but you're getting almost no detail. But then look at the right. That's the same sample viewed with phase contrast. All of a sudden, everything inside just pops with this sharp, dark contrast. So, how does it do that? Well, the secret is this really clever bit of physics called phase shift. So, imagine light waves marching along in perfect step, right? When they pass through denser parts of a cell, like the nucleus, they get slowed down just a tiny bit. They fall out of step with the other waves. Now, our eyes can't see this tiny timing difference, this phase shift, but a special kind of microscope sure can.
So, how does this magic actually build an image? Let's walk through it. First, a special diaphragm shapes the light into a hollow cone. As that cone of light passes through the cell, some of those light rays get slightly delayed. That's our phase shift. Now, here's the genius part. A special phase plate inside the objective lens intercepts only those delayed rays and slows them down even more. So when all the light waves finally recombine, the twice-delayed waves are now perfectly out of sync with the others. This causes them to interfere and basically cancel each other out. And just like that, an invisible timing difference is turned into visible darkness and contrast. It's an absolutely brilliant solution.
But what if you don't just want to see the structure? What if you want to identify specific materials, or maybe even tag individual molecules? Well, for that, scientists develop techniques that use the properties of light itself to make things glow and shine in amazing ways.
You know, one of the coolest methods is polarized light microscopy. It's designed to see specific materials like crystals or collagen, things that have this unique property where they can split a beam of light in two. By using a pair of polarizing filters, the microscope makes these structures light up in these vivid, beautiful colors. And this isn't just for making pretty pictures, it's a super powerful diagnostic tool. A doctor can identify the painful urate crystals that cause gout in a patient's joint fluid simply by seeing their characteristic bright glow under the scope.
Okay, now let's talk about making things glow in the dark, because that's basically the essence of fluorescence. Certain molecules, called fluorochromes, have this amazing property. You hit them with high-energy light, say blue light, and they absorb it, and then they spit that energy back out as a different color, like green or red.
So scientists took this idea and ran with it, creating fluorescence microscopy. They can design these fluorescent dyes that act like tiny beacons, and they attach only to the specific molecules they want to study, like a certain protein or a strand of DNA. The microscope then uses filters to shine only the exact color of light needed to turn on those beacons.
A special mirror reflects that light onto the sample, and then only the glowing light from those tagged molecules passes back through for you to see. You're literally painting parts of the cell with light to track where they go and what they do.
For all its power, though, light microscopy has a fundamental physical limit. You literally can't use light to clearly see things that are smaller than about half the wavelength of that light. For centuries, this diffraction barrier meant the tiniest structures, things like viruses, just stayed as blurry mysteries. To see them, we had to abandon light completely.
And this is the result. The electron microscope. We're talking a potential magnification of up to 1,000,000 times or more. That's about 1,000 times greater than the best light microscopes. This wasn't just an improvement, it was a total revolution that opened up the entire world of ultrastructure, the incredibly fine details inside our cells.
So how does it pull this off? Well, the core difference is fundamental. A light microscope, right? It uses photons, particles of light, and it focuses them with simple glass lenses. An electron microscope swaps out light for a beam of electrons, which have a much, much shorter wavelength, and it replaces glass lenses with powerful electromagnetic fields. That's the key that allows it to resolve structures that are smaller than a single nanometer.
And believe it or not, the story doesn't end with electrons. In just the last few decades, Nobel Prize-winning techniques called super-resolution microscopy have found these ingenious new ways to break that old diffraction barrier, but by using light. These methods essentially let scientists watch individual molecules at work inside living cells in real time. It's like combining the detail of an electron microscope with the dynamics of a light microscope. The age of discovery is far, far from over.
Wow, okay, we have covered a lot of ground, from simple glass lenses all the way to powerful electron beams. So let's just do a quick recap of the right tool for the right job.
This table is a great little summary. Bright field, that's your basic everyday tool for looking at stained tissues. Phase contrast, like we saw, is the hero for observing living, unstained cells. Fluorescence lets us track specific molecules by making them glow. A more advanced version of that is confocal, which uses a clever pinhole to block out-of-focus light, letting scientists build these stunningly sharp 3D images. And finally, of course, the mighty electron microscope, which uses electrons to see the truly tiny stuff, like viruses. Each one is a specialized tool for asking a different kind of question about the world.
You know, from Hooke's cells all the way to seeing the dance of individual molecules, our ability to see has completely defined our understanding of biology. We've pushed the boundaries so far that we can now visualize individual atoms. And it just leaves you with this final, tantalizing question. What other invisible worlds are still out there, just waiting for the next great invention to finally bring them into the light?
Thanks for joining us on this amazing journey into the unseen world. If you enjoyed discovering how we see the microscopic, definitely be sure to subscribe for more videos like this.